Now we're moving on to genetic engineering. So what is genetic engineering first of all? When genes are altered in a microorganism to code for a desired product. One of the most famous examples here is modifying genes in a bacterial cell so that it produces the hormone insulin, which can be used to treat diabetes type 1. You need to know the names of two very specific enzymes. Number one is the restriction enzyme used to cut DNA. Number two is the ligase enzyme used to join the cut ends of DNA back together. Another good word to know the meaning of is the term vector. A vector is used to transfer biological material from one organism to another. A good example is that bacterial plasmid I just described in insulin manufacture. Another example is the mosquito, which transfers the plasmodium to the human host. A plasmid, remember, is a small circle of DNA found in bacterial cells. And now we're ready to describe in detail how bacterial cells can be genetically engineered to produce human insulin. So remember, human insulin is needed to lower our blood sugar levels after we've eaten a meal. It converts glucose to glycogen. For people with type 1 diabetes, they're unable to make insulin. Historically, they used to use pig insulin, but for all kinds of reasons such as ethics, the fact that the human would reject that pig insulin, it was deemed an unsuitable method. Genetic engineering provided a way in which we could make huge amounts of insulin without the need of injuring any pigs, and it would be perfectly matched for the human recipient. So number one, a restriction enzyme is used to cut the human insulin gene from a healthy person. The same restriction enzyme is used to cut open the bacterial plasmid. A ligase enzyme is used to seal that insulin gene into the bacterial plasmid, and the sticky ends, which are exposed ends on the DNA, are sealed together. They are complementary to each other. Because the plasmid now contains human DNA, it's known as a recombinant plasmid, and as I previously mentioned, the bacteria is acting as a vector here. The bacteria is then placed in a fermenter, which is a large vessel with optimum conditions to ensure that that bacteria replicates as quickly as possible to make huge quantities of human insulin. That insulin is then purified and is ready for use. As well as genetically modifying bacteria to produce hormones like insulin, it's also possible to genetically modify plants. So now let's run through a specific example. So for example, say we want to genetically modify a plant so that it produces more beta-carotene. Remember, beta-carotene is a precursor of vitamin A, and it helps our eyesight and enables us to see more clearly in low light conditions. Hence the expression, carrots help you see in the dark. Carrots are full of beta-carotene, so therefore good for your vision. So how do we genetically modify a plant so that it can produce beta-carotene? Well, first of all, we need to use a restriction enzyme to open up that bacterial plasmid, and the bacteria we use is known as an agrobacterium. Restriction enzymes are also used to isolate the desired gene in the target DNA. We know that sticky ends will be produced because that will enable the ligase enzyme to seal the desired gene into the bacterial plasmid. And again, we have what's known as a recombinant plasmid. The recombinant plasmid is inserted into the agrobacterium bacteria, and leaf discs from the plant are treated with the recombinant agrobacterium. The leaf discs are cultured in a nutrient-rich medium in sterile conditions, and any plantlets that grow will have that desired gene, and in this way they're known as transgenic plants because they contain a gene from another species. So you might want to know, why is agrobacterium used here? Because agrobacterium is often found freely in soil, and it is known to introduce plasmids into plant material, making it the perfect bacteria for this process. So we've already described that using genetically modified plants can increase their nutritional value, such as this example here we said, which meant that more beta-carotene was made. Other reasons we might genetically modify plants is to increase their shelf life, to make them more resistant to frost, to make them resistant to herbicides, weed killers, so that when the farmer adds weed killers to his crops, you're not going to kill the actual crop that you're trying to grow. It's even been known that tobacco plants have been modified to produce hepatitis B antigens. This has potential use in creating a vaccine in order to prevent hepatitis B infection. So now we're going to outline the advantages and disadvantages of genetically modifying plants. So the main advantage is that you might create higher yields because of reduced competition from weeds, as well as reduced damage by pests. It's possible that crops can be grown in more extreme environments, meaning that there's less famine due to more crops being grown. Tied in with one of the points I've already made, genetically modified plants mean that less herbicides and less fertilizers need applying, which makes it cheaper and easier for any farmers. and obviously much better for the environment. And as I've already previously mentioned, genetically modified plants increase the nutritional value to humans. They have a more complete combination of lipids, proteins, vitamins, minerals, and I've mentioned beta-carotene on several occasions. But what are the disadvantages of genetically modified plants? Well, they could cross-pollinate with weeds, creating super weeds which are hard to kill using herbicides. They tend to be expensive, and you find that poor farmers can't afford genetically modified seeds. And it also reduces biodiversity by decreasing the species of weeds and pests, meaning there's less food available for consumers higher up the food chain.